In my group, the main research is focused in the fabrication of silica and Silsesquioxane-based materials. We have been working with mesoporosilica nanoparticles, developing different structures with different sizes, different pore sizes that allow us to carry different type of cargos, mainly for biomedical application. In a similar way, we have developed Polysilsesquioxane nanoparticles where we have derivatized different approaches for the multifunctionalization and multimodal approach of these nanoparticles. All these results has been published in different papers by my group, and today, as I was mentioning, I will focus on our recent or current progress in the synthesis and application of Polysilsesquioxane nanoparticles. So Polysilsesquioxane nanoparticles is a great platform to use as a delivery system. We strongly believe, that's what we are doing the research with this platform, that we can multifunctionalize the system and create multimodal approaches using these platforms. For instance, we can incorporate drugs, photosensitizer, or stimuli-responsive system or prodrugs incorporated inside of the nanoparticle. We can also functionalize the nanoparticle, the exterior surface of the nanoparticle, with imaging agents, or target or incorporate also nucleic acids in the surface of the nanoparticle. Further, we can modify the system with polymers to change the chemical or physical-chemical properties of the nanoparticle with certain features to improve the interaction with biological entities. And we can also create target systems using antibodies or other targeting agents that will allow to render that property to this platform. We have design engineered this platform in such a way that we have a high loading capacity. We can render biodegradability and biocompatibility to the system. The main application in my group for this Silsesquioxane platform, Polysilsesquioxane platform, or PSilQ, the way that I abbreviate the name, has been in cancer treatment, either by using individual therapies or by combining with different type of therapies. One of the main focus of my group in terms of the clinical goal has been the treatment of breast cancer, which as we know, breast cancer is a challenging disease. And in particular, we are directing our efforts in a type of breast cancer that is called triple negative breast cancer. This triple negative breast cancer, or TNBC, due to the biological features of the cancer, in particular the lack of very specific biomarkers, make it very difficult to treat it. And even when it's treated with regular regimes like chemotherapy, one of the main results is actually making the disease worse, meaning it usually creates resistance to those treatments and resulted in metastasis. So there is a really clinical need, urgent clinical need of coming up with different therapies or alternative therapies to treat this disease. Our group and others believe that the combination of different therapies can be a promising alternative to improve the ultimate outcome of the treatment of this disease. In my group, recently published the development of Polysilsesquioxane nanoparticles, where we integrate 3 different types of therapies: chemotherapy, photodynamic therapy, and gene therapy. To do that, we incorporate curcumin as an anti-cancer drug, protoporphyrin 9 as a photosensitizer, and nucleic acids for gene therapy. So the engineering approach that we have developed in my group to synthesize this type of materials, of Polysilsesquioxane materials, is by functionalizing the building block or the active component in the nanoparticle with tricolciclane functional groups that are used for polymerization that will help us to grow the nanoparticle and for the main framework, which guarantees a high loading capacity of these active components. In addition, we also add what we call stimuli-responsive linkers that will allow us to, under specific stimuli like pH or high-reducing environment, it will allow us to break down the nanoparticle, as I will show you in the next slides in the second project. And we also add functional groups that are used for further modification of this material. As a proof of concept of this idea, of this approach, we use protoporphyrin 9. Protoporphyrin 9 is a well-known photosensitizer agent that is clinically approved. So we chemically modified protoporphyrin 9, adding the features mentioned already mentioned, where we add tritoxysilane, which again is gonna be used or is used for polymerization to grow the network, the framework of the nanoparticles. We add a disulfide bond, which is our stimuli-responsive linker, which responds to high-reducing environments such as that found in cancer cells. And we have carboxylic acid, which are used for further functionalization of the nanoparticle. So by using a microemulsion approach, we fabricated the nanoparticles using the building block as described before, with protoporphyrin 9. We can have two versions just with the protoporphyrin 9, or we also have the combined version where we co-condensate protoporphyrin 9 and curcumin. These nanoparticles is further functionalized with polyethylenimine to render a positive charge in the nanoparticle. And we also functionalize by using the carboxylic acid that I mentioned before, and with polyethylene glycol. Finally, the positive charge of the polyethylenimine, PI, help us to load the nucleic acid by electrostatic interaction. So we characterize these nanoparticles with a wide variety of tools. I will show some of the key results that we obtained for this nanoparticle. TM showed that the nanoparticle is fairly spherical with an average diameter of 40 nanometers. TM-SEM support that result. And by analyzing the material using dynamic light scattering, we can find the hydrodynamic diameter is slightly higher than the diameter that we found by TM, most likely to some slight aggregation to due to the hydrophobic properties of these nanoparticles, the presence of the porphyrin there. Zeta potential fairly characterized the different functionalization of the nanoparticles, going from negative to positive charge, depending of the presence or not of PEI or PEG. We characterize the phototherapeutic properties of this system using MDA MB231 cells, which are TNBC cells. By inoculating these cells with the materials and irradiated with light for the phototherapeutic effect, we found out a typical dose response curve for the cell viability of the cells. By determining the IC50, which is the amount required of material to kill 50% of the cells, we found out that there is a synergistic effect between the combination of protoporphyrin 9 and curcumin. And another interesting result that we found out is that the toxic effect of the nanoparticles in the dark condition is lower than the protoporphyrin 9 by itself. So it's reducing the toxicity in dark or cytotoxicity in dark of that of the photosensitizer. So we further tested the capability of this system for silencing for gene therapy. In this particular case, we use MDA MB231 cells that express green fluorescent protein. We loaded our nanoparticles with siRNA that knock down that protein, the expression of that protein. And by using flow cytometry, we determine the silencing of our nanoparticles. We can see here that there is the silencing is concentration dependent. The higher the amount of nanoparticles, higher the silencing. And we also found out that there are an effect of the surface properties of the nanoparticles. The presence of PEG actually reduced the silencing effect. By using fluorescent microscopy, we actually confirmed the data that we got from flow cytometry. As you can see here, compared with the control, our material reduced or knocked down the expression of green fluorescent protein very effectively. So we move on to the application of this system for in vivo settings. Using an orthotopic model of TNBC. For this presentation, I will focus on two main parts or three main parts of the research, which is the degradability of the system, both in solution and in vivo, and also the toxicity and the phototherapeutic effect of the platform. And why I'm focusing on these three main components, because those are key components to translate or the success in these three components are determine the success for the translation of this platform for clinical applications. So as I mentioned before, the building block contains, the protoporphyrin building block contains a disulfide bond, which is broken under reducing environment. That is the main component of the system that will allow us to enhance the biodegradability of the nanoparticle. So we tested in solution using DLS by looking at the hydrodynamic diameter of this platform under a reducing environment. At different times, we can see that there is an increase on smaller particles under the again determined by DLS as an indication that the nanoparticles start to degrade in comparison with a control nanoparticle that there is not a reducing agent in the solution. And also not only the reducing environment increase the degradation, but also the release of the photosensitizing agent, in this case protoporphyrin 9, as we can see here, where we determine the release of PP9 after being triggered by the presence of the reducing environment. We can see the stimuli response of the system. We move on to test the biodegradability and excretion of our nanoparticle in vivo using immunocompetent mouse model. For this experiment, we functionalized the nanoparticles with near-infrared dye to determine the luminescence of the material inside of the animal, and we determine, or we focus on two main regions: the abdominal region of the animal and the bladder region, to show the degradability effect of the system. We can see that the kinetics of accumulation in the abdominal region reaches a maximum at certain period of time, in this case 4 hours, and start to decrease. In the case of the bladder region, the accumulation of the material start to increase and keep increasing as an indication, or that's what we hypothesize, that the material is being excreted through the renal excretion pathway. We determine the biodistribution of our nanoparticle after 10 days of administration of the material, which, by the way, is administered through intravenous injection. We focus on two main components of the nanoparticles, which is silicon and protoporphyrin 9. And we tested 3 different concentrations of the nanoparticles and compared with a control, which is an animal without any administration of the material. And we also use a mesopure silica nanoparticle as a control of a silica system that is not degraded as easy as in the case of the PSilQ nanoparticles to compare the difference. So by determining the amount of silicon using ICP-OS, we can see that clearly or nanoparticle, PSilQ nanoparticle, there is a minimal accumulation of the silica in the main organs, liver, kidneys, and lungs. And in comparison, again, as I was mentioned, as a positive control, which is MSN, that we can see that the silica is accumulated in lungs and liver. This data was further confirmed by looking at protoporphyrin 9, that the protoporphyrin 9 in the different organs are the same at the same level as the control, as an indication that there is not an accumulation of the nanoparticles, of the components of the nanoparticles, and has been excreted from the animal. So we move on to test the therapeutic effect in an orthotopic model of TNBC. So this is the treatment, the treatment regime for the experiment. We have 5 injections, again, intravenous injections of the material at different times with a period of 4 days between injection, and 3 different light irradiations, 6, 12, and 24 hours after the injection. By measuring or plotting the tumor growth, we can we determine the effect of the treatment, and we can see clearly that the nanoparticle that has been, the PSilQ nanoparticle that has been irradiated with light is the one that showed the highest reduction in the tumor burden for this model. This was further confirmed by weighing the tumor, by determining the tumor weight after sacrificing the animals. We see that this platform is the one that presents the best therapeutic result. By using ex vivo analysis, in this particular case of the tumor, using immunohistochemistry, the tunnel assay, by looking at apoptosis, we can clearly see that the group of animals that were treated with the PSilQ nanoparticle in the presence of light are the ones that show the highest population of apoptotic cells. Toxicity, we look at the toxicity of the system. We didn't see any toxicity, even looking at the decrease in weight during the whole treatment, or also looking at histopathology. There is not any adverse effect. So finally, biodistribution of the system show that well, something that is particular for nanoparticles that change the biodistribution of the drug. So in this particular case, we can see that in red, which are nanoparticles, there is no accumulation of protoporphyrin 9 in the main organs, different to the group where the protoporphyrin 9 was injected by itself. We can see accumulation in these organs. And by looking at silicon, we also determine that indeed the nanoparticles are accumulated in the tumor. So just by summarizing my presentation, I hope that I have convinced you that this PSilQ platform is a great platform for the drug delivery, where we can combine different type of different type of therapeutic modalities. Like I demonstrate you, we can combine photodynamic therapy with chemotherapy and gene silencing. And by tuning the properties of this system, we can enhance the loading capacity of the agents in the nanoparticle, creating a synergistic effect for the treatment of TNBC. We also show in vivo that by this approach of having reducing a linker or stimuli responsive linker, we enhance the biodegradability of the nanoparticle, but still keeping a good therapeutic effect or phototherapeutic effect in the orthotopic model of TNBC. So finally, I want to acknowledge my group, the members of my group, in particular those that directly work in this project, Dr. Ridima Juneja, Dr. Mubin Taranun, Dr. Zachary Lyles, and Priya Badaribu. And last but not least, I would like to acknowledge the funding agency that supports the work that we are doing in my group. Thank you for your attention.